Now that we know a neuromuscular junction is essential to cause our skeletal muscle to contract, let's look at some of the proteins of muscle contraction. So if we're looking at the anatomy of a skeletal muscle fiber, just a reminder that when we refer to a skeletal muscle fiber, we are talking about a skeletal muscle cell. So a skeletal muscle fiber is a cell, and you can see here in the image this is a cell and it has multiple nuclei and striations, which you'll see striation source are dark and light alternating bands of contractile proteins. We can also see there are smaller organelles within this skeletal muscle cell or fiber, as well as little mitochondria. Sarcoplasm is our name for the cytoplasm inside of the cell, and the sarcolemma is the plasma membrane. Notice there is a myofibril. The muscle fiber is made up of small contractile organelles known as myofibrils. So the muscle fiber is a cell and the myofibril is an organelle. Those myofibril organelles, notice, are made up of different contractile filaments that, again, essentially are responsible for these dark and light bands, the striations of the muscle cell. So myofilaments are the proteins, contractile proteins, that make up the organelle myofibril. There are two major types of contractile myofilaments. They are the myosin filament, which is also known as the thick filament, and the actin filament, which is also known as the thin filament. And in this image, you can see that they've shown the thin filament to be here in the red and the thick filament to be in the blue. And so we'll look a little bit more at the structure of these filaments, because notice they always go into patterned bands, and these patterns are known as sarcomeres. So the sarcomeres are the smallest repeating functional unit inside of our muscle cell. So a muscle fiber, again, is a cell, a myofibril is an organelle, and these myosin and actin filaments are contractile proteins inside of the myofibrils, that organelle. So if we look more closely, notice that here they're showing these red-looking branches as our myosin or our thick filament, and then these little yellow lines are going to be our actin filaments. And we're also going to see there are some other proteins that are supportive, but the actin and myosin are the major contractile proteins. We see that there are sarcomeres, again, our smallest contracting unit of our muscle, and they are labeled by these bands and lines. So we've got an H zone, Z disc, M line, A band, and I band. Let's look more closely at these. So we are going to name those bands based on how they make up the sarcomere. So the ends of them are bounded by what we call Z discs. But notice the actin and myosin are arranged where they have dark and light areas based on overlap. Where you just have actin or myosin without any zone of overlap, there are light-colored bands. When you have zones of overlap of actin and myosin, they are dark-colored. So under a microscope, a sarcomere would have these striations where they alternate light, dark, where it's overlapped, light, where it's just myosin, dark, where it's overlapped, and light, where it's just actin. And as a muscle contracts, notice that these bands on the sarcomere get smaller. So notice that we can see these light, dark bands when we have a relaxed muscle in this first figure. Here we see a partially contracted muscle in the second figure. Notice that our bands of overlap, our dark bands, are getting longer. And then in a maximally contracted muscle, our bands of overlap, or the dark bands, are much larger. So let's look more closely at the structure. So a sarcomere is bounded by Z discs. I think of them as zigzag lines. Z disc to Z disc always is a sarcomere. Right in the middle, there is a middle line, so this is the M line. This bisects the sarcomere. In the center, the dark line, it's a light band actually, so we see back here, it's a light area because it just contains myosin. In the middle, that is our H zone, so the H zone just contains the myosin filament. The A band travels the length of the entire myosin filament. So if we look how it's labeled here, it goes the length. So it includes the area where we just have myosin that's light in the middle, but also these two dark areas where we have an overlapping area of myosin and actin. So A band goes the entire length of the myosin filament. H zone just goes on the area where there's no overlap. And finally, I bands bisect two different sarcomeres because there's a Z disc in the middle, and they cover just the area where we have actin, so no overlap. And as we partially contract a muscle, our H zone and our I bands get smaller. And in a maximally contracted muscle, we have very minimal, if any, area on I band, and our H zone completely disappears because what's happening are our actin filaments are pulling in toward the M line, and our zone of overlap is getting bigger. So we have no more H zone, which was just myosin filament. And if all these contractile units, if all these sarcomeres within a myofibril do this at once, then you have complete shortening and contraction of the muscle fiber. There are some other proteins involved in our sarcomere. We know our contractile proteins are myosin and actin, but we're also going to talk heavily about some regulatory proteins, troponin and tropomyosin. And you're going to see these involved more when we watch the video of the cross-bridge cycle. So they are heavily involved in muscle contraction in the cross-bridge cycle. Troponin and tropomyosin regulate muscle contraction by being involved in our actin filament. So notice here in yellow, this is our actin filament. Tropomyosin is this brown line, and what it's doing is it's covering what we call active sites on actin. So you see these little yellow globular units, that's called globular actin. They're these little proteins, but they've got these little dark colored areas that are covered by that tropomyosin. And then you also have this little, looks kind of like a blue flower, these three blue areas that are also on the actin filament, and that is troponin. So I think of it like this. If you are trying to get into my house, you may have to open a door. So if I am the active site on actin, if you want to get to me, you may have to open my door. So tropomyosin is the door. If I don't want you to come in my house, unless I invite you in, then I'm going to have a lock on my door. So troponin, the little blue area here, is like the lock. You have to have a key to unlock the door. So we will see in the cross-bridge cycle video that calcium is the key that changes the shape of troponin that will allow tropomyosin to roll away from the active site on actin. So calcium is the key, unlocks troponin, which is the lock, pushes open the door, which is tropomyosin, and that exposes active sites on actin. Note that muscle contraction in the cross-bridge cycle, which we'll watch eventually, cannot happen without tropomyosin rolling away from the active sites on actin and exposing those active sites. We'll also look here at the structure of myosin. It looks like two woven golf clubs with these heads. And these heads are ATPases, meaning that they can break down or hydrolyze ATP and release energy. And when these myosin heads release energy, we see these ATP binding sites, they can pivot back and forth between a 90-degree angle, so they can move. And they also have an actin binding site. So these myosin heads can bind to these actin active sites when tropomyosin is rolled away. We're also going to see a couple structural proteins within our sarcomere. They're a little less important for our detailed purposes of the cross-bridge cycle, but we'll look at titin, nebulin, alpha-actin, myomesin, and dystrophin. So if we look a little bit more at the detail here, you're going to look at this kind of light-colored white line that I'm moving my mouse along. This is titin. This is going to anchor the thick filament to the M line. Notice the M line is made out of what we call myomesin. If I go to the more detailed structure of actin, so again, here are globular actin units. It's just a different drawing. You've got the yellow parts, which are representing the active sites. This blue woven line, which is tropomyosin, that's the door covering the active sites. These brown jelly bean looking structures, which is troponin, that is our lock covering or keeping tropomyosin in place. And then running through the middle to stabilize our or align, our actin is nebulin. It's an inelastic protein that helps align the thin filaments. And alpha-actinin, this is going to help make up our area where we have the Z discs here. So that's going to be where alpha-actinin is. Finally, we also have another structural protein known as dystrophin. And what dystrophin does is this is going to transmit the tension of our sarcomere shortening to the sarcolemma so that we can transmit that tension and have muscle contraction. There are some defects in this dystrophin protein in people that have muscular dystrophy, which causes muscles not to work properly. So we'll look at the cross-bridge cycle next, and we'll see how these proteins, particularly our contractile and regulatory proteins, contribute to contraction of muscle.